Welcome to the PubMed Daily Review for April 10th, 2026. Today we scanned 7,948 new open access papers. Here are the top 5 selected for detailed analysis. Up next, our first paper out of 5, Tumor lactate metabolism, shapes, immune suppression, and therapeutic resistance revealed by integrative multi-omics and digital pathology by Bohai Fong and Johan Hess from Artificial Intelligence Research Center, Zhejiang Laboratory of Medical Epigenetics, Department of Cell Biology, School of Basic Medical Sciences, Hangzhou Normal University, Hangzhou, China. Let's break down its key contributions. Cancer cells reprogram their metabolism to survive and grow, and lactate production is a major part of this. This lactate doesn't just fuel the cancer. It also creates an environment that suppresses the immune system and makes tumors resistant to treatments like immunotherapy and radiation. Currently, measuring this lactate activity is complex and costly, limiting its clinical use. This research sought to find a simpler, more accessible way to assess tumor lactate metabolism directly from standard pathology slides. To tackle this, the researchers first identified a set of 59 genes related to lactate metabolism. They then integrated multi-omic data, including gene expression and spatial information, with digital pathology. Using this, they trained a deep learning model to predict whether a tumor had high or low lactate activity, simply by analyzing standard H&E stain slides. This model was tested across various cancer types and validated in an independent patient cohort. The results were quite promising. The deep learning model developed from H&E slides showed strong performance in distinguishing between high and low lactate activity tumors, with AUCs ranging from 0.73 to 0.82 in HNSCC and even higher, up to 0.89, across other cancer types. Tumors predicted to have high lactate activity were indeed associated with faster growth, less immune cell infiltration, and worse responses to immunotherapy and radiation. Importantly, this digital prediction was validated by showing higher levels of key lactate-related proteins, LDHA and MCT1, in the predicted high lactate tumors. This study presents a significant advancement by demonstrating that we can assess a tumor's metabolic state, specifically its lactate activity, using readily available digital pathology images. This offers a practical and scalable tool for identifying patients likely to have an immunosuppressive tumor microenvironment and potentially poorer responses to current therapies. Ultimately, this metabolism-informed digital biomarker could help personalize cancer treatment by guiding patient selection for specific therapies or combination strategies. Up next, our second paper out of 5, Transamniotic Fetal Delivery of Cystic Fibrosis, CF, Transmembrane Conductance Regulator, CFTR, mRNA by Camilla Moskowitz-Sova and Dario Faza from Department of Surgery, Boston Children's Hospital/Harvard Medical School, Boston, Massachusetts, USA. Let's break down its key contributions. Cystic fibrosis is a genetic disease that can lead to serious complications at birth, like meconium ileus. This is caused by a faulty CFTR protein. We explored a new idea. Could we deliver the instructions, or mRNA, for making the correct CFTR protein directly to the fetus while it's still developing? The goal is to see if this transamniotic route could be a way to prevent or treat these early life complications. To test this, we used a rat model. On gestational day 17, we injected a specially prepared CFTR mRNA, wrapped in a protective lipopolyplex, directly into the amniotic fluid of all fetuses. We compared this to a control group that received the injection without the mRNA. We then examined the fetal intestines and lungs at different time points up to term to see if the human CFTR protein was present. The study showed that nearly all the mRNA was successfully encapsulated. Fetal survival was high and similar between groups. Crucially, we detected human CFTR protein in the fetal small intestine of the mRNA group at all tested time points, with statistical significance. Interestingly, CFTR was also found in the lungs, but this was also seen in the control group, suggesting possible species homology. No human CFTR protein was found in the maternal blood. These findings suggest that delivering CFTR mRNA directly into the amniotic fluid is a promising new approach for potentially treating cystic fibrosis-related meconium ileus before birth. The protein appeared in the right location in the fetal intestine, which is a critical step. While exciting, Future research must confirm that this delivered protein actually works and demonstrate its effectiveness in animal models of cystic fibrosis. Up next, our third paper out of 5, mGem, opening ENV and harnessing NK cell effector functions to eliminate HIV when infected cells, by Jonathan Richard and Andres Finzi from Centre de Recherche, Duchu, Montreal, Quebec, Canada. Let's break down its key contributions. Despite effective HIV treatment, the virus hides in long-lived cellular reservoirs. To achieve a cure, we need new ways to eliminate these hidden viral sanctuaries. One exciting approach is to boost the immune system's ability to kill infected cells, a process known as antibody-dependent cellular cytotoxicity, or ADCC. This paper explores a novel strategy using 2 key components. First, small compounds called CD4MC are designed to force the HIV envelope protein into an open state, exposing vulnerable parts of the virus. Second, type I interferons are used to increase the amount of viral envelope on the cell surface and enhance natural killer, or NK, cell activity. Together, these aim to make infected cells more visible and susceptible to immune attack. The study demonstrated that CD4 mimetic compounds effectively forced the HIV envelope into a more exposed conformation. Concurrently, type I interferons successfully increased viral envelope proteins and BST2 on the cell surface, while also diminishing the virus's ability to evade the immune system via PO. This combination significantly enhanced natural killer cell effector functions, leading to improved recognition of infected cells. In summary, this research presents a compelling strategy combining CD4 mimetic compounds and interferons to potentially reduce the HIV-1 reservoir. By enhancing antibody-dependent cellular cytotoxicity and NK cell activity, this approach could significantly improve immune recognition of infected cells. While promising, further investigation is crucial to determine its clinical efficacy and pave the way for a functional HIV cure. Up next, our fourth paper out of 5, Post-mortem evidence of pathogenic angiogenesis and abnormal vascular function in early Alzheimer's disease, by Daniel J. Aspey and J. Scott Miners. Let's break down its key contributions. Alzheimer's disease is a devastating neurodegenerative disorder, and we know that problems with blood flow and the integrity of the blood-brain barrier are involved. But how exactly do these vascular issues contribute to the buildup of amyloid and tau proteins that characterize AD? This research aimed to shed light on this complex relationship by examining brain tissue from individuals with varying stages of Alzheimer's pathology. To investigate this, the researchers analyzed brain tissue samples from individuals across a spectrum of Alzheimer's disease severity, from healthy controls to those with advanced pathology. They measure a range of biochemical markers related to blood flow, vessel integrity, and the hallmark proteins of AD. By comparing these levels across different disease stages and brain regions, they sought to uncover the intricate connections between vascular health and neurodegeneration. The study revealed significant findings. Cerebral perfusion, as indicated by the MAG/PLP1 ratio, was already reduced in the early stages of AD. Markers of vascular dysfunction like VEGF, EDN1, and CD31 were elevated in these early stages and were linked to this reduced perfusion, while BBB leakiness, measured by fibrinogen, was more pronounced in late-stage AD. Evidence of pathogenic angiogenesis was present even in the early stages. These results paint a picture of dynamic vascular changes occurring throughout the course of the disease. This research highlights that vascular problems aren't just a consequence of Alzheimer's, but appear to be involved from the early stages. This opens up exciting possibilities for new therapeutic targets, such as interventions aimed at controlling EDN1 and VEGF signaling to protect the brain's vasculature. While this study provides crucial insights, future research should continue to unravel the complex interplay between vascular health and AD progression. Up next, our fifth paper out of 5. Telomere damage enhances immunogenicity of neuroblastoma and accelerates response to anti-PD-L1 treatment, by Stephanie Hopner and Matthias Fischer from A Department of Experimental Pediatric Oncology and Hematology, University Hospital Cologne, Faculty of Medicine, University of Cologne, Cologne, Germany. Let's break down its key contributions. Neuroblastoma, a common childhood cancer, often has a poor prognosis, especially in high-risk cases. Current treatments are intensive, and new strategies are desperately needed. While immune checkpoint inhibitors like anti-PD-L1 have revolutionized cancer therapy, neuroblastoma is often resistant. This study investigates if we can wake up the immune system within these tumors by inducing telomere damage. To test their hypothesis, the researchers used a genetically engineered mouse model that mimics aggressive neuroblastoma. They treated these mice with 6-thio-DG, a drug that causes telomere damage, and an anti-PD-L1 antibody, both individually and in combination. They then meticulously tracked tumor growth, survival, and analyzed the molecular changes within the tumors to understand how the treatments worked. The results were promising. Mice treated with the combination therapy showed a significant delay in tumor growth and a notable increase in survival compared to those receiving either treatment alone. Importantly, the combination therapy overcame the initial lag phase often seen with anti-PD-L1 treatment alone. This was linked to the activation of the C-gas-sting pathway by 6-thio-DG, leading to increased PD-L1 expression and a greater infiltration of immune cells into the tumor. This study provides compelling preclinical evidence that targeting telomere damage can enhance the effectiveness of immune checkpoint blockade in neuroblastoma. By activating the C-gas-sting pathway, this approach may help overcome the immune resistance characteristic of this aggressive childhood cancer. These findings pave the way for exploring this novel combination therapy in future clinical trials, offering hope for improved outcomes in patients with high-risk neuroblastoma. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.